This is part 2 of the lecture from chapter 20. We are still on viruses. And just real quick, I'm going to read the different hypotheses on how viruses evolved, how they originated. You don't have to learn these, but this is just for your information. But proteins and nucleic acids, which are the components of viruses, the organic molecules that are found in viruses are proteins and nucleic acids. And these evolved in living cells as well, and viruses may have arisen from these two basic polymers when cells did. Viruses may have been derived from pieces of cell genomes. Viruses may have evolved backwards from living cells. Viruses could have degenerated from living cells. But in any case, we do know a lot more about the reproduction of viruses. We know a lot more about that than we do about the evolution of viruses. And the reproduction of viruses has 5 basic steps, no matter which virus that we are looking at or talking about. The first step is always attachment. The second step is penetration. The third is called biosynthesis. And then maturation and release. So in the first step, attachment, the virus has to attach itself to the host cell. And this is going to be based on a match between the molecules on the surface of the virus and the host cell receptors. Okay? In the next step, the host cell is going to either engulf the virus, or the virus is going to inject its genome into the host cell. That would be penetration. And then the biosynthesis step is when the host cell is used to synthesize the new viral components, which would be the nucleic acids, the proteins, any of the enzymes that are part of the virus itself. Then all of those components are assembled into new viruses. That is the maturation step. And then the new viruses will be released from the cell. And sometimes this process will kill the cell, and sometimes it won't. It will just damage it. So those are the steps of viral reproduction. Now, we know a lot about the life cycle of bacteriophages. And I told you earlier, a bacteriophage is a virus that infects bacteria, viruses that infect bacterial cells. And these are important for a lot of reasons, but one of the main reasons they're important to us is because we have studied them. And we've learned a lot about virus behavior from these viruses that infect bacterial cells. So there are 2 life cycles that we've learned about with bacteriophages. They are the lytic cycle and the lysogenic cycle. In the lytic cycle, basically all these steps that we just learned happen. Okay? So the virus, at the end of the lytic cycle, hundreds of virus particles are released. The host cell undergoes lysis, in other words, it bursts, and releases hundreds of virus particles. This happens in the lytic cycle. The lysogenic cycle is a cycle that allows the virus to remain dormant for oftentimes many, many years. And this dormancy or latency, it's known as latency, allows that virus to remain dormant or latent or inactive for a period of time until conditions are better for the virus to begin reproducing again. So, if we go up here to the beginning of this, viral reproduction does not occur immediately but may occur in the future. The virus becomes integrated into the host genome, the viral DNA or RNA becomes integrated into the host genome. And this period of time where the DNA of the virus or the RNA is integrated into the host genome is known as latency. And the latent viral DNA is called a prophage. And we can say DNA, it doesn't have to be, I keep saying DNA or RNA, but bacteriophages are DNA viruses. So we can actually just say DNA. And the viral DNA that's incorporated into the host genome is known as a prophage. Diphtheria is a human disease caused by a prophage-carrying bacterium, which produces a toxin that damages the lining of the upper respiratory tract, restricting breathing. So even though diphtheria is a bacterial disease, it is caused by a bacterium that is infected with a virus, with a bacteriophage whose DNA has been incorporated into the bacterium. So that's a very interesting case, right there. So the lytic cycle is the active cycle, and the lysogenic cycle is the dormant or latent cycle. Now, as far as the reproduction, the steps of viral reproduction, this is a large blown up picture where you can see the virus is the green structure, and you can see the attachment part where it has attached itself to the host cell, in this case a bacterium. So the structures in the virus have attached to receptors on the host cell. And then the penetration step, in this case, the virus injects its DNA into the host cell. And the biosynthesis stage, because what we're looking at is the lytic cycle or the active cycle, the host cell is now tricked into producing the components that are required to make new viruses. So we see the green capsid and we see the purple DNA. And then the maturation is incorporating or assembling all those components into new viruses, mature viruses. So the DNA is now inside the capsid, and those viruses are ready to be released and infect new host cells. And that is the next step, is the release, in which the bacterium, the host cell, is actually destroyed. And those new viruses, and it's only showing 5 here, it's normally in the hundreds. Those hundreds of new viruses now can go infect new bacteria, bacterial host cells. So here we have the 5 steps of the lytic cycle, and now for the lysogenic cycle. If the virus it goes into the lysogenic cycle, it will do that after penetration. So after penetration, integration happens when the viral DNA is incorporated into the host cell DNA, and that's now called a prophage. And the host cell will remain unharmed. It just will contain the viral DNA, and it will remain latent until conditions improve for the host cell to reproduce, and the host cell and the virus will now enter into the biosynthesis step of the lytic cycle. So the lysogenic cycle only happens when the virus is actually going to remain dormant for a time before it reproduces. It will also not cause any symptoms during this lysogenic cycle. It will only cause symptoms when it is in the lytic cycle. So lytic cycle means active virus, and lysogenic cycle means inactive or latent or dormant. All right. For animal viruses, it's very, very similar to the reproduction of bacterial viruses, like the lytic and the lysogenic cycle. We've learned a lot by studying bacterial viruses or bacteriophages. But the animal virus is going to enter the host cell and release its viral DNA or RNA, and Budding is a process where viral particles are released in a bud, which is like, we learned about this. It's called exocytosis. So it's very similar to exocytosis. And just like in exocytosis, the viral particles will be now surrounded by the cell membrane or plasma membrane of the host cell. And so they'll now have an envelope that is very similar to the cell membrane of their host cell. Now, retroviruses are very special because, like an example of a retrovirus is HIV, human immunodeficiency virus, which is the virus that causes AIDS. These viruses contain an enzyme called reverse transcriptase. Now, we have learned that what happens during transcription, which is the first step in protein synthesis, is that DNA makes a copy called messenger RNA. But these viruses are RNA viruses. And so what happens is the RNA copies itself into DNA, cDNA. This is reverse transcription, when RNA actually copies itself into DNA, and that DNA can become integrated into the host DNA. And as the host DNA replicates, so does the viral DNA. And this is kind of like the lysogenic cycle because it allows that virus, such as HIV, to remain latent for sometimes for years. After a while, the viral DNA is transcribed into RNA again. New viruses are produced, we say the virus has become active. So this is how the HIV works, which is a retrovirus. Okay, so you can see, first of all, the capsid is the green structure here. You can see that there's RNA as the core, and then there's a green capsid, and outside there is an envelope, and the envelope contains these glycoprotein spikes that match up with the receptor on the host cell. In this case, it would be a helper T cell in humans. And so that's the attachment stage, and then we have the entry of the virus by endocytosis. And then we have reverse transcription, where the viral RNA makes a copy of DNA. That DNA incorporates itself into the host DNA. It's now called a provirus. It's incorporated into the host DNA. And then after a period of time, it can be years, it can be days, but after a period of time, the DNA undergoes transcription and translation just like it would in any cell. That's the biosynthesis stage, and the components of the virus are now synthesized. And then the maturation is where those components are assembled. The virus is going to have a capsid, it's going to have RNA as its core, and then that, as it is released by exocytosis, it actually obtains its envelope, which is a portion of the host cell membrane, and that enables it to trick a new host cell into thinking that it's harmless. Okay, the flu virus has two spikes. It has an H spike, and these spikes, remember, are glycoproteins. It's got an H spike and an N spike. H stands for hemagglutinin, and N stands for neuraminidase. The H spike allows the virus to bind to the receptor. There's 16 different H spikes. And the N spike attacks the host plasma membranes. There are 9 different types of N spikes. So each type of spike can occur in different varieties. Our immune system only recognizes H and N spikes that it has been exposed to. Currently, we have the H7N9 and H5N1 subtypes of the flu that are a great concern because they can potentially become pandemics. This shows you an illustration of how bird flu and human flu can integrate. So here is the human influenza virus, and you can see the orange and the pink